Good afternoon, everyone. My name is Karolina Cieminska. I'm from Magdalena's podcast laboratory group from University of Gdańsk. And today I'd like to tell you something about molecular aspects of thermostability of TS2631 endolysin that derive from phage TSC2631 that can infect Thermus cathoductus bacteria. Okay. We have to wait when slides start moving. I'm trying. Okay. Okay, it's working. Okay. So we can all agree that we live in era of multi-drug resistant bacteria. And we are looking for new strategies to fight back them. And at first, I think we should find the target for our drugs. So one of the options is peptidoglycan. Peptidoglycan is a structure that is built of glycan strands that are cross-linked by short peptides. And there are some similarities and differences between species. For example, in most peptidoglycan in gram-negative bacteria, in the third position of stem peptide, we can see mesodiaminopimelic acid, contrary to gram-positive bacteria, where there is present lysine. But there are also some exceptions. For example, Thermus thermophilus is an gram-negative bacteria, and in third position of stem peptide, there is ornithine. Okay, but what is the common part of all of these proteins, peptidoglycans? Is the presence of one residue, is alanine that is present here in the first position of stem peptide. And this is very well conserved structure. And this is why we should consider peptidoglycan as a target for our drugs. Okay, but we can't skip one more thing. In gram-negative bacteria, there is a present outer membrane, contrary to gram-positive bacteria, where is no layer on the peptidoglycan. So if you want to find drugs that target in peptidoglycan in gram-positive bacteria, we don't have to be worried. But in case of gram-negative bacteria, we have to find drugs that can pass through outer membrane of negative bacteria. Okay, so what characterizes a good drug? First of all, it should not disturb any physiological process. It shouldn't be harmful to the patient, but harmful to the pathogen. And of course, it should have the minimum side effects and should be highly stable, resistant to proteolysis and chemical denaturation. And today we will focus on the last part, what describe very well thermostable proteins and why proteins can be stable at higher temperatures. First of all is their hydrophobic core, and that increased compactness of protein. And here we can see example of thermophilic endolysin. And on the right, you can see the mesophilic and hydrophobic parts are depicted in red. Second of all, it's metal ion binding that actually works only at higher temperatures. And here we can see the active center that is coordinated by zinc ion. Also presence of some residues, for example, disulfide bonds that are and salt bridges that are created by cysteine, and for also presence of proline. They can adapt only to few configurations, and that decreases the entropy of protein unfolded state. Okay, another one is arginine that has here the guanidium-like head group. And because of it, it has reduced chemical reactivity and can interact with charged ions and residues. And another one is tryptophan and other aromatic residues that can interact between themselves. For example, it is an aromatic interaction that takes place when between two phenyl rings, space isn't higher than 7 angstroms. And another one is cation P interaction, and it takes place when positively charged residues interact with the center of phenyl group. Okay, we also observed that in thermophilic endolysins proteins is more tryptophans, prolines, and arginines. And here we have an example of thermophilic endolysin TS2631 that has more, for example, more tryptophans, 3.5 more than in mesophilic enzymes. Okay, so let's focus on endolysins. It is able to lyse bacteria after external application, and some of them can pass through outer membrane of gram-negative bacteria. It hydrolyzes peptidoglycan, so it is what we are looking for. And endolysin that derive from phages that infect gram-positive bacteria mostly have modular structure and are built of enzymatically active domain connected by linker with cell wall binding domain. And they can also have more than one of each domain. And example of such endolysin is lambda SA2. And endolysin that derive from phages infecting gram-negative bacteria mostly has globular structure and are built of enzymatically active domain, but there are also parts that are responsible for interacting with peptidoglycan. An example of such endolysin is the S2631. Okay. So we are looking, they are potential antimicrobial factors, and I will repeat that they should be highly stable, resistant to proteolysis and chemical denaturations. So enzymes like this, we should search among thermostable microorganisms. And the object of our studies is actually endolysin TS2631 that derive from thermophilic bacteriophage TS2631 that was isolated on Iceland from hot springs and can infect Thermus scotoductus and Thermus thermophilus. It's an amidase. That means that it cleave bond between sugar moiety and stem peptide. And it is this endolysin is coordinated in active place by zinc ion, has length 156 amino acids, and its molecular weight is 18 kilodaltons. What is unusual for this protein is that it has N-terminal flexible region with 7 positively charged residues, and because of this, this protein can pass through outer membrane of gram-negative bacteria. And it was checked if this endolysin can degrade some species of order Enterobacterales and Pseudomonadales. And we found out that actually is active against multi-drug resistant Acinetobacter baumannii and Pseudomonas aeruginosa. And this is a very good news because actually in Europe, in almost half countries, we had at least 25% Acinetobacter species that were resistant to carbapenems. Okay, so this endolysin can degrade peptidoglycan. We know that. And how it works. And the optimum temperature for this enzyme is 60 degrees Celsius. And here in panel A, we can see the native cell. And here after treatment of TS2631 endolysins. And the same experiment was repeated on Acinetobacter baumannii at 37 degrees Celsius. And here also we can see that there is native cells And we can see that this endolysine works also at 37 degrees Celsius. Okay, in our laboratory, we have 3 more thermophilic enzymes, and only one FICO has melting temperature lower than 100 degrees Celsius. And the melting temperature of FICO was obtained by differential scanning calorimetry, and 3 other melting temperatures of 3 other proteins were obtained by nanodifferential scanning fluorimetry. That is a lot of more efficient for these studies. Okay, so we asked ourselves what makes the endolysine TS261 so thermostable, and can we obtain more stable mesophilic structural homologs of TS261 endolysine? Today we will partially answer to the first question. Okay, we have performed site-directed mutagenesis, and we substitute with alanine all tryptophans, prolines, and arginines, and also residues that are responsible for catalytic center and residues that are conserved. But today I will focus only on prolines and tryptophans substitutions variants. So at first, nanodifferential scanning fluorimetry analysis. We could observe that only one protein variant, proline substituted at position 140, had significantly lower melting temperature. And but this is not so easy actually to say what melting temperature really matter in that way. So we have to perform, of course, more experiments. We did also to measure the activity of this enzyme turbidity reduction assay. We had protein that we were heating at 90 degrees Celsius by 15 minutes, and we weren't heated. We added the substrate that was thermophilus, and after incubation of that proteins with substrates at 60 degrees Celsius by 15 minutes, we obtained their relative activity. And we also could observe that proline at position 140 after heat treatment at 90 degrees Celsius has lower relative activity. And that results overlapped with nanodifferential scanning fluorimetry analysis. Okay, so we have here we have tryptophans variants, and among 7, 3 of them has significantly lower melting temperature. It was tryptophan at position 102, 109, and 145. We also did turbidity reduction assay in the same conditions, and we also saw that tryptophan at position 102, 109, and 145 had significantly lower relative activity to wild type enzyme after heat treatment. But what we also observed that tryptophan at position 53 has lower relative activity even at 60 degrees Celsius. But melting temperature of this protein is very similar to wild type enzyme. So it interested us, and we wanted to perform more experiments, especially for this variant. But firstly, short summary. So we know that tryptophan at position 102, 109, and 145, and also proline at position 140, are involved in protein thermostabilization. But what is the function of tryptophan at position 53? So at first we thought it can be some change in conformation, but we see that wild type enzyme is an alpha helix, and tryptophan at position 53 doesn't really change its conformation. So we decided to perform one more experiment. At first it's peptidoglycan binding. So at first we had to isolate peptidoglycan from thermophilus. After that, we incubated a buffer with peptidoglycan and protein. And after centrifugation, we obtained 2 samples with supernatant, where was unbound protein, and pellet, where was peptidoglycan with bounded protein. And here we can see carbon anhydrase. It is our negative control that should be only in unbound fraction. And we could see that our variant tryptophan 53 is present also in unbound fraction. It means that it binds peptidoglycan. So to sum up, tryptophan at position 53 takes part in peptidoglycan binding and is present here, far away from the core of protein. And tryptophan at position 102, 109, 945, and also proline at position 140 are involved in protein stabilization, and they are all present on the C-terminal of endolysin. Okay, so what are our future plans? We of course want to perform more analysis for other variants. And what is more, like I said before, we want to check if it is possible to substitute proteins, residues that are responsible for thermostability in TS2631 in their mesophilic homologs. For example, we find one enzyme that is a homolog. It is an endolysin from Klebsiella pneumoniae bacteriophage, K11GP3.5. And yes, we are looking for more enzymes like this. Okay, and at the end, I would like to acknowledge to our foundators and of course my all co-workers, my group, and especially my supervisor for helping me in everything. Okay, and a little advertisement at the end. So antibiotics has a special issue and wait for 15th of December 2022. So if you want to publish something, you can consider antibiotics. And if you have more questions, so Magdalena Płotka is here, that is guest editor. And after conference, you can ask her about something. So thank you a lot for attention.
So, having worked out what makes that particular endolysin thermostable, can you then take, for example, another endolysin and change some residues to more tryptophan? Are you intending to try to apply what you've learned in that endolysin to other ones?
You mean other thermostable endolysins or mesophilic?
Yeah, just perhaps a mesophilic one to make it more thermostable.
Yes, yes, and there was an example of that one, that K11, I didn't say that this is a mesophilic endolysin. Yes, and we want to find out if we can make it more stable. Yeah. And we're looking for more endolysins like this. We're looking for homologs because then we can actually do this.
Sleepy, but
okay. Thank you.
that was really lovely insights into how to make something a lot more effective. So thank you very much, Karolina.